Good morning and welcome to the video for meiosis and the meiosis paper crossing over activity. You will need your notebook open to page 97 with these notes on them. You also need the 2 pages of chromosomes cut out, so you will need 3 like that, cut out like this, and then these 3 cut out like this. You will also need some paper clips and then your paper crossing over lab. So the first thing I'm going to talk about really quickly is just to go over what meiosis is. So we talked about mitosis, which makes your body cells, and it makes an exact replica of your body cells. So if your skin cell does mitosis, you get another skin cell. Meiosis is different in that it creates your sex cells, which are called gametes. So it takes a stem cell and it either makes an egg if you're a woman or a sperm if you are a man. So your body cells are called somatic cells, and this is what does mitosis. So in mitosis, just as a reminder, you have, as a human, you start with 23 chromosomes. You go through prophase, metaphase, anaphase, telophase, cytokinesis, and then from there you get 2 identical daughter cells. They are exactly the same cell as this parent cell. This is not a stem cell. This is a skin cell or a heart cell or an eye cell or something, okay? So 2 identical cells, and these are referred to as diploid cells or diploid cells, and that di means 2, so you've got 2 is like a pair of chromosomes, okay? Like a pair of shoes. Now, on the other hand, meiosis makes your sex cells, or the gametes, okay? And that's the only kind of cell that they make. It starts out very similar to mitosis. You have a cell, and it goes through mitosis, and it creates 2 identical cells. But then what it does is it does mitosis again, so you end up with 4 cells that have half the number of chromosomes as the original cell, okay? So mitosis ends in 2 cells and meiosis ends in 4 cells. Ms. Whiteley once pointed out that you can also think of it like meiosis because it makes sex cells, okay? There are 4 different cells. They are called haploid cells, and like you think of the like half, so they have only 1 set of chromosomes instead of the pair. Okay, so what we're going to do is we're going to model this process using paper chromosomes, and to keep it from being too long and terrible, we're only going to do 3, 2 regular chromosomes and the sex chromosomes, okay? So you're going to look at your lab, and you should have all of these materials. The male and female chromosomes cut out, the paper clips, and then if you go to this tiny URL, it'll take you to a website that allows you to roll a dice. And you only need to roll 1 die, okay? If you need to pause the video while you go do all that stuff, now's a good time to do it. All right, so in the introduction, it goes into an explanation about how the body cells of plants and animals are diploid. So 2N means, so N is like a chromosome, and so 2N means you have 2 sets of chromosomes, so you have the pairs, okay? And then haploid just is the N because it's only half the amount of chromosomes, right? Gametes, which are called egg and sperm, are haploid cells. When they join together at fertilization, they make a diploid cell. So you take half plus half, put them together, you get the 2, and that's when you are called a zygote, okay? So it's 2 successive divisions of the nucleus, and then mitosis only has the 1. All right, so you're going to cut out your chromosomes. Already did that. And now you're going to have to do the first phase, which is interphase. And in interphase, there's an S phase. And during S phase, your chromosomes are replicated. So what you're going to need to do is on all of your chromosomes, you need to replicate your chromosomes. So no active gene. I'm just going to put none, none. Okay, brown hair, which is the capital B. It's important that you copy it exactly because they are exactly the same. Height, which is a capital H. This is none. This is melanin production, which is a capital M. The gene to make amylase, which is the enzyme in your spit, is also a capital A. And then there's a none down here. So you go ahead and pause the video and fill in the rest of your chromosomes and unpause when you have completed that. All right, now that you have your chromosomes filled in, I would like for you to look at chromosome number 1 for a second from both of them, okay? So you see here how the chromosomes are the same. The spaces are the same. There's no active gene, no active gene. Here's a hair color gene. This is the height gene. There's none. This is melanin, which gives you the color of your skin. Amylase, which again is an enzyme in your spit, and then none. You don't have active genes on all of your DNA strands. And when they do something called genome mapping, the order that they're in for humans is all the same. So every human on Earth, right in this chromosome number 1, this third spot, would have the gene for brown hair right here. It's just the different combinations of dominant or recessive that give you the different hair colors. So every human on Earth, if these were real, which they're not, everyone would have this brown hair color gene here, and the height gene would be right here. Okay, so you have your chromosomes copied. That was S phase. Congratulations. Now we are going to perform mitosis. So first we're going to your original DNA looks like this. Oh, I'm sorry. You have one from your mom and one from your dad, okay? Then we did S phase, so now we've replicated, and now we have 2 sets of all of our DNA. And this is a boy because he's got an X and Y chromosome here. All right, so now we have our 2 sets. So now we're gonna go into M phase, which is where the nucleus goes poof, and all of our chromosomes are released. Then we're going to go to M phase, in which they line up in the middle, and I know you're not going to be able to see all of this, but that's fine. I'm going to line them up in the middle, and then we're going to do anaphase, where they're pulled apart. And by pulled apart, we mean cut them apart, okay? We're going to separate our chromosomes. In your body, this would be done by some spindle fibers. They're like ropes that attach to these middle parts right here that are called centromeres, and it would just yank them apart, like pulling apart Velcro. And they would split, and half is going to go to one side and half is going to go to the other side. So you have chromosome 1 splits apart. This chromosome 1 also splits apart. Chromosome 2 splits apart. This chromosome 2 also splits apart. Here's our X chromosome for our gamete, splits apart. And then our Y chromosome for the gamete splits apart. So now you have 2 new nuclei. So if you drew a nucleus around this and a nucleus around this, these would be your 2 new nuclei for telophase. Now, meiosis is different in that it doesn't stop there. So we divide, now you've got 2 cells. And now we're going to immediately go into another prophase, metaphase, anaphase, telophase. But first, your body does something a little strange in which it recombines the genes, kind of like if you wanted to mix up a salad, so you shake it. That's kind of what your body does, except instead of shaking, it just cuts things and pastes them onto other things. So this is where you're going to need your dice roll. You're going to roll the dice, let's say for chromosome 1. And you're going to roll once for above and once for below. So, for instance, you need to get a 3 or lower to cross over your chromosomes 1. So if you get a 6, you just keep rolling until you get a 1 or a 2 or a 3. So I rolled my dice and I got a 2. So I'm going to count down 2 chromosomes. And I'm going to cut here. I'm going to cut here. And then I'm going to switch them. I'm going to get my tape. And I'm going to tape them together. Okay. All right, and then I'm going to roll my dice again for the bottom. So I've got 1, 2, 3, 4, 5 down here. So you have to have a 5 or lower. So I rolled a 2. So now I'm going to count. Let's see, is it from the top or the bottom? Let's look at our 3 squares before the centromere. Yeah, we'll start from the bottom. So I got a 2. So I'm going to go here. I'm going to cut off these bottom 2. This is how we get genetic variation. This is why you don't look the same as your brother or sister, and why sometimes in your family, like, you can look a lot like your aunt or your uncle. I have a niece and she looks just like me. She's just like me, but she's not my kid. Okay, so now we have crossed over chromosome 1. Now you're going to repeat this process for chromosome 2 and the sex chromosomes. And then once you're finished, you're going to paper clip them together at their centromere. Okay, like this. So now they're paper clipped together at the centromere. Once you're finished with this, the crossing over, then you're going to repeat PMAT. So we just did, this was prophase. Now we're going to go through metaphase again, where you line your pairs up in the middle, and then anaphase, where they get split apart, and then telophase, you'll end up with 4 new cells. Okay, so this was our first pair of chromosome 1. This would be our second pair, which you also have to do crossing over for, and this would split apart. And these are going to be your 4 separate cells. Okay, so then you'll separate them, stack them each 4. So then at the very end, you'll choose one of your 4 cells for the next part. Do not throw these away once you're done. Once you have your 4 separate cells, I would like for you to paperclip your whole stack together so that you keep all your chromosomes for 1 sperm or 1 egg together. And you're going to answer these questions on the back. And once you have done that, you're going to get a piece of white paper. If you're a girl, you're going to make an egg. And if you're a boy, you're going to make a sperm. You're going to choose one combination of your things, and you're going to put them on your gamete, okay? And then this is going to be for the next part. So you're going to choose one entire set of whatever, okay? Not that. So you need a chromosome 1, a chromosome 2, and a Y or an X chromosome. And then if you're a boy, you're going to do the same thing on your sperm, and you just tape them on there. And then follow the directions in the next video.